Welcome to Bioratna. Dear students, I am Chandana Sengupta and our today's topic of discussion is the process of transcription from the chapter Molecular Basis of Inheritance. The process of transcription. In my previous video lecture, I have discussed the transcription unit separately as it needs special attention. So please go through it before seeing this lecture. Let us start with what is transcription. Transcription is the process of copying of genetic information. The genetic information is present in DNA. So it is the process of copying. It is the process of copying of the genetic information. Genetic information from one of the strand of DNA. DNA has two strands. So from one of the strand of DNA into RNA. Not both the strands are not involved. Both the strands are not involved. The DNA strand which is going to form the RNA, only its one part is to be transcribed. Means one strand is having many genes and one gene is being transcribed into RNA. This much clear? Thus, we can write it like this: DNA which is forming RNA. So this process is called transcription. Now this RNA is of three types. What are the three types? One is ribosomal RNA, one is transfer RNA and one is messenger RNA. What is the role of rRNA? This rRNA, it has structural and catalytic role. It has structural and catalytic role during translation, where the proteins will be formed as per the information which is present in mRNA. What is the role of this tRNA? tRNA, it carries the amino acids. It carries the amino acids to the site of translation. Also, it reads the code. It reads the code present on mRNA. And what is mRNA? This is the messenger RNA. It is the messenger of DNA. It has copied the message from the DNA that what will be the sequence of amino acids in the protein. So mRNA is the messenger which has the information for the formation of proteins. All these three are required for translation. Now the question arises, there are three RNAs, rRNA, tRNA and mRNA. So is there one enzyme or there are separate enzymes which help in the formation of these three different enzymes? In prokaryotes, it is only one enzyme, RNA polymerase, DNA-dependent RNA polymerase, which does all the three functions, means formation of ribosomal RNA, tRNA and messenger RNA. Whereas in case of eukaryotes, it is a specialized one. There are at least three different RNA polymerases which form these three different types of RNAs. Other than this difference, there are few other differences between prokaryotic and eukaryotic transcription. Mostly it is similar. There are very few differences. Let us see what are they. Though the steps of transcription are similar in prokaryotic and eukaryotic organisms, but there are few differences in prokaryotic and eukaryotic transcription. What is that? In case of prokaryotic transcription, it takes place in cytoplasm. Why in cytoplasm? Because they do not have nucleus and the DNA is present in cytoplasm. Whereas in case of eukaryotes, process of transcription, it takes place in nucleus for the simple reason that DNA is present in nucleus. Second difference, in case of prokaryotes, they need one initiation factor. Start करने के लिए only the main enzyme, that is RNA polymerase. DNA dependent RNA polymerase is not sufficient. It cannot start initiation. Start नहीं कर सकता है. So it needs the sigma factor. This is the initiation factor, and rho factor. This is the termination factor. So it needs sigma as well as rho factor. Whereas in case of eukaryotes, they do not need sigma or rho factor. Fine. Now another difference, one major difference. The third important difference is that in case of prokaryotes, it is the single enzyme, only one enzyme which forms all the three types of RNA, that is ribosomal, messenger and transfer. Whereas in case of eukaryotes, there are three different enzymes in the nucleus. And what do they do? If it is the enzyme number 1, remember the keyword R, M and T. This R. Enzyme 1 will form ribosomal RNA, enzyme 2 will form messenger RNA, and enzyme 3 is going to form the transfer RNA. Then there are further finer detailing that you will do, but first मोटा मोटी इसको ऐसे याद रख लो. Now this one RNA polymerase 1, it is going to form ribosomal RNA. It forms different types of ribosomal RNA. They are 28S ribosomal RNA. Ribosomal का R छोटा लिखा जाता है. Then 18S ribosomal RNA and 5.8S ribosomal RNA. What is this S? This is the Swedberg unit. Swedberg was a scientist. When the different components, organelles, the molecules of a cell in a centrifugation machine, during centrifugation, when they settle down, the time taken by them, that is, for that the unit is the Swedberg unit. So this is S for Swedberg, this small r is for ribosomal. The second one, it forms messenger RNA, but this messenger RNA, it is not a mature one. It is immature messenger RNA which needs further processing, and it is called heterogeneous nuclear RNA. This is called heterogeneous nuclear RNA. Now the last one, enzyme 3, it forms the tRNA. So it forms transfer RNA, tRNA. It also forms 5S ribosomal RNA and small nuclear RNA. So these are the three different types of enzymes which are present in the eukaryotes. Is there any more difference? Yes. In case of prokaryotes, the next step of transcription, that is translation, It starts immediately means even when the process of transcription is not completed, then only it starts, means process of translation starts. Why? There are two reasons. Translation takes place in cytoplasm, and the RNA formed, they are already in cytoplasm in case of prokaryotes, whereas in case of eukaryotes, they are present inside nucleus, then they will come out of the nucleus and then they will carry out translation in the cytoplasm. The second point is that the mRNA which is formed in case of eukaryote, it needs further processing, whereas prokaryotic mRNA, it does not need any processing and it can immediately act as a template and it can immediately start the process of translation. So these are the few differences between prokaryotic and eukaryotic transcription. Now let us see what is a transcription unit. The two strands of DNA, one strand and this is the second strand. Let us write its polarity. Now that part which is to be copied into mRNA, it is called structural gene. So this part is structural, this is the structural gene. The transcription unit which we are seeing, it has three parts. One is structural gene. Second is the promoter. What is promoter? Promoter is the sequence of nitrogen base. यहाँ सारे DNA के sequence ही तो है nitrogen base के. So it is also having a nitrogen base sequence which is recognized by the enzyme RNA polymerase and it attaches at this place. So now it will move, it has to copy the information in structural gene. So naturally it has to move in this direction and here there is terminator which says stop. So here the process of transcription stops. So this is promoter, this is structural gene and this is terminator. Out of these two strands, whose information will be copied? That one will be called template. कौन है template? This one or this one? The one which is having its 3' end towards promoter. I repeat because this is very important. That strand which is having its 3' end towards promoter, it is called the template strand. So this one is the template strand. So it is this strand which is template and this strand which is the coding strand. This coding strand is displaced and it does not take part in transcription, still it is called coding. The sequence in the coding strand are exactly the same as in the newly formed mRNA except that thymine is replaced by uracil. Let us take one example. Suppose this is the template strand and this template strand, it is having the sequence A, T, C, C, G, C. Then in case of your coding strand, it will be A, here it is T, A, G, G, C and G. In case of mRNA, this is A will pair with U, then A, G, G, C and G. You can see here that the only difference is this, in place of T it is U, whereas A, G, G, C, G, here also A, G, G, C, G, both are the same. So you see that the sequence in mRNA and in coding strand, they are the same. Now what? Now let us see why the enzyme is called DNA dependent RNA polymerase. RNA polymerase. RNA न्यूक्लियोटाइड को ये पॉलीमराइज कर रहा है ना, so naturally it will be called RNA polymerase, and for that it needs the help of the template strand. It will see what sequences are there in the template strand, and then this enzyme will accordingly by following the complementarity rule, it will put these nucleotides of RNA. That means it is dependent on DNA. That is why it is called DNA dependent RNA polymerase. That's it. Now one more point, then, that is the substrate of transcription. You will say that it is easy. It is the nucleotides, the RNA nucleotides, क्योंकि ये nucleotides का ही तो polymerization है. So it is the nucleotide which are the substrate. No, it is not so. Nucleotides are required, but those nucleotides, like in case of replication also, they are carrying with them the energy required for the process. So if I draw one nucleotide, this is the oxygen, and then this is the pentose sugar I am drawing. This pentose sugar, at one position there will be nitrogen base. It may be A, it may be U, it may be C, or it may be G, any one. Then on this 5 position there is this phosphate group, so you call this nucleotide. Polymerization of this nucleotide will occur, but here there are 2 more phosphates attached. Fine. So this is called, in phosphate को अगर हम अलग से बोलते हैं, this is nucleoside, sugar plus the nitrogen base, that is nucleoside. So this is nucleoside, and these are the 3 phosphates, that's why it is triphosphate. This is nucleoside triphosphate. Done. When these bonds they are broken, then that will release the energy which will be used for the process of transcription. So now you are ready to understand different steps of transcription. Now we are going to see the different steps of transcription, that is initiation, elongation and termination. The different steps, student, the diagrams I have not drawn beforehand, because as students they find the diagrams difficult. So let me see whether it can be made bit easy. So let us start. So the first, what you will do, draw. I will take one step extra than NCERT. That will make your understanding easier. The first one, you draw simply the 2 strands of DNA. Drawn. Now this is the another strand. So this is the DNA helix. This is 3. This is the 3 prime, and this is its 5. This is 3 and this is its 5. Fine. So what is this? This is DNA helix. You yourself can decide. You take your own structural gene. चलो यहाँ से start करते हैं और यहाँ पे ख़त्म करते हैं. ठीक? So you can visualize on this side it is the promoter and this side it is the terminator, and this much to this much it is the structural gene. Now the DNA helix, like in case of replication also, replication में पूरा DNA एक साथ तो open नहीं हो गया था, यहाँ भी ये part by part open होगा. तो for the 3 steps, initiation, elongation and termination, we will show 3 different parts opening up. पहले हम यहाँ से यहाँ तक का दिखा देंगे, फिर यहाँ से यहाँ तक का दिखा देंगे. So that will be our diagram. Let us do it. See, now what we are doing, ऐसे लिया इसको और फिर यहाँ से इसको open up किया. So ये open up हो गया, इतना open है, और फिर से इसे जैसे आपने हेलिक्स बनाया था वैसे ही हेलाइकल स्ट्रक्चर दिखा दीजिए. Process starts from 3' end na. So if we are starting from here, so this strand, this is the 3' end. This is initiation. This is initiation. मतलब अभी start हो रहा है transcription. Let us not draw the details here. First only let us draw the 3 diagrams having the 3 different parts opened up. So now the next one here, अब इसे बंद कर दीजिए, इसे बंद कर दीजिए और यहाँ पे ओपन कर दीजिए. So now this one will become, ये बंद हो गया and this has opened up. Again this is the closed one. Now come to, this is the elongation step. This is elongation step and the last step is the termination step. In termination हम ये लास्ट पॉइंट तक पहुंच जाएंगे, यहाँ तक पहुंच जाएंगे. सो नाउ अगेन व्हाट यू शो, यहाँ से यहाँ तक का काम पूरा हुआ और ये ओपन अप हो गया. सो दिस इज द क्लोज्ड वन, हियर इट इज ओपन एंड दिस इज इट. Here you show the nitrogen bases, here also you show the nitrogen bases. Now let us start. The first step is initiation. The RNA polymerase, it attaches to the promoter and then it starts, these are the nitrogen bases, उसका complementary base वो A, U, C, G डालना शुरू कर देता है. So here we are showing the RNA polymerase. This is DNA dependent RNA polymerase. It cannot start the initiation on its own. तो वो मदद किसकी लेता है? वो मदद लेता है सिग्मा फैक्टर की. This is the sigma factor. Sigma factor and together they are called holozyme. Holozyme, the complete enzyme. Now it can start the initiation. Done. This much it will do. Then this one will close up and the next part will open. This RNA polymerase itself, it can go on opening the DNA helix. So now it has moved to this side. It has moved to this side. The sigma factor, जैसे initiation start हो जाता है, then the sigma factor is no more required. It falls off. तो here the sigma factor is lying ना. शुरुआत करने के immediate बाद sigma factor has fallen off. Now RNA polymerase has moved ahead. तो इसने जो अकेला है ना, सिग्मा फैक्टर नहीं है उसके साथ, यहाँ से लेके यहाँ तक का उसने mRNA बना दिया. तो let us draw this mRNA. This is the mRNA. Then it will move. This one will close when this much mRNA is made. Then this will close, and the next part will be opened up. And here, when it reaches this side. When it reaches this side, then due to the termination factor here, the termination factor, it is the termination factor which helps it in termination of the process. So then the termination factor, the RNA polymerase and the messenger RNA all fall off. This is when we are showing the rho factor as well as the sigma factor. That means this is the prokaryotic transcription. When we don't show sigma factor or rho factor, then it is the eukaryotic transcription. So let us summarize this one again. See, this is the DNA helix. From here to here we have shown the structural gene. The RNA polymerase, it attaches to the promoter. It cannot start the initiation by itself, so it attaches to sigma factor. Then sigma factor falls off and the RNA polymerase moves ahead, opening the DNA helix. सामने से वो खोलते जाएगा DNA helix को, और the part where it has already transcribed, that will close. So this one has closed. It has moved ahead. It has formed this mRNA. Then it moves to the termination site, and then all the three things, that is RNA polymerase, the Rho factor and the RNA, they all fall off. Fine. So this was all about transcription. Hope it was clear to you. If you have liked the video, then do subscribe for many more such video lectures. Thank you for watching the video.